Hello, and welcome to the fifth webinar in a series about ocean sewage pollution. My name is Kristen Mays. I'm the Reef Resilience Network, your host for today's webinar. The Ocean Sewage series is presented by the Nature Conservancy with support from the Reef Resilience Network and NOAA's Coral Reef Conservation Program. During today's webinar, Understanding Sewage Impacts in Hawaii, we'll hear from 3 experts about how sewage pollution is being identified and documented in Hawaii and how that information is being used to address sewage issues. Dr. Kim Falinski from the Nature Conservancy Hawai'i will share about community-scale research to understand wastewater impacts and which areas should be prioritized for management action. Next, we'll hear from Dr. Dan Amato from the University of Hawai'i at Mānoa, who will present about statewide efforts to map impacts of sewage pollution and guide cesspool removal. And finally, we'll hear from Michael Mezzacapo from the state's Cesspool Conversion Working Group, who will share about the group's efforts to incorporate state-of-the-art science into a planning and decision-making process. As a reminder, the Reef Resilience Network is developing resources for managers to address the threat of sewage. So in addition to this webinar series, we're creating web pages on the network toolkit, case studies highlighting sewage monitoring and management strategies, and an online course to help managers build understanding on this topic. We're excited. The web pages are in their final stages of development and will be published in January, so stay tuned. A couple quick housekeeping items. Today's webinar will be 1 hour long. Following the 3 presentations, there will be a discussion session. There are 2 ways you can ask questions. You can type your question into the question box at any time during the presentation or during the discussion session, of course. Or you can raise your hand during that session and we'll call on you, go ahead and unmute you, and then you can ask your question out loud yourself. If you're having any technical difficulties, such as trouble hearing or seeing the slides, please send us a message via that same question box and we'll do our best to resolve the issue. Before we get to the presentations, we'd love to hear from you via a quick poll. So we'd love to know if you're currently working on water quality. And so by this, we mean, are you doing water quality monitoring, research, or have any projects that are planned or underway to improve water quality? Give you a few minutes. Wow, okay, looks like most of you are currently working on water quality. Only 2% are unsure, and no one picked this topic. Scares me. So this is really encouraging, also very surprising that we have that many folks involved, which is wonderful. And that is a great tee-up. It's just great to know who's on the other side and who's joining us for the webinar today, so we appreciate it. We will go ahead and hear from Dr. Kim Falinski to start. Go ahead and give you a moment to get set up, Kim. And as a reminder, Kim is joining us from the Nature Conservancy's Hawaii program, and she'll be sharing about community scale research to understand wastewater impacts. Hi, Kim.
Hello.
We can see your slides, just if you can pop them in the presenter mode.
Great, that's what I was looking for. Are we all good? Everybody can hear, you can see my slides?
Good to go.
Thank you. Thanks so much for having me onto this webinar. I really appreciate it, Kristen and Reef Resilience Network, and I look forward to being able to share some tidbits about what we've been doing here in Hawaii, working with communities to identify coastal water quality issues specifically related to sewage. I have a lot of co-authors on this slide because we work with many, many partners across the state and across our marine program here in Hawaii. So thanks to everybody, and you'll see their names coming up throughout the presentation. So even though Hawaii is known for its iconic beaches, we do have concerns here for cesspools. We have about 88,000 on-site disposal systems throughout the state, which discharge about 55 million gallons per day of sewage. And that means on our 4 major islands, we have numbers in the thousands: 50,000 on Hawaii Island, 14,000 on Kauai, 11,000 on Oahu, 12,000 on Maui. And if you don't know, cesspools, which in Hawaii were developed in the early 1900s here and used for a long time, are often dug close enough to the groundwater that they're directly connected to the ocean, leading to possible coastal contamination. And even some of our nicest beaches, like the one on the right at Hanauma Bay, that one notoriously had one in the past and has upgraded since, but there are lots of other of our beach parks that do still have cesspools. My role at the Nature Conservancy and the Nature Conservancy's role throughout the state is to best support communities who are interested in maintaining their coastal resources. And in this case, it means thinking about what types of land use practices are affecting their water quality. And we've really focused on developing research partnerships with academic institutions to be able to help solve this question. We have state partners like the Hawaii Department of Health that are federally funded by the Beach Act, which provides money at that level to be able to do regular coastal monitoring. We have citizen science initiatives that we partner with to be able to work with communities themselves who are interested in taking on this issue themselves. And then there are, of course, nonprofits like the Nature Conservancy Coral and many, many, many others that can be used as facilitators to work for these research needs and communicate the results to their communities. So across Hawaii, I'm going to focus this talk on 3 different communities that we're working in here. One of them is Maunalua Bay in urban East Honolulu. It's quite urbanized place. It has a lot of its streams are gray streams, and that is one of our communities. We work with Leeward West Maui, and I specifically have worked with a group called Hui o Kawai Ola, which is a citizen science-based initiative to help work on water quality issues there. And then Puako. Puako is a relatively undeveloped coastal community in West Hawaii that has a number of cesspools and has been really the focus of some pretty intensive work to think about what are the best ways to upgrade, especially when you have a volcanic substrate. So on our newest island of Hawaii, it's a different substrate. And all of our partners were listed above, and I'll keep mentioning them. So when I think about water quality and think about water quality monitoring and what you really want to do here, you have to start with that interest. The community has to be interested in thinking about what their problems might be. And that is the start to the problem. From there, you can look at what is the initial, how do you start water quality monitoring, which site selections do you do, just to figure out what the story is. Do you have problems? Do you not have problems? And that can involve experts helping you design the study. And then and then advanced studies. So from there, you might be able to then follow up and find the more specific sources that are problems. After you do all of that, you still have data analysis and communications projects that can feed into a planning process that would help use that coastal water quality to inform on-land actions. And in this phase 1, which I'm thinking about, I think of these 4 different parameters that you can test for. And if a lot of you have done water quality work, maybe some of you have touched on some of these different parameters. So you have chemical parameters like nutrients and silica, physical parameters like dissolved oxygen and turbidity, bacteria, so you can use different fecal indicator bacteria like Enterococcus, and then phytoplankton. Some people measure for chlorophyll A because if you know the response of the phytoplankton community, you can try to determine what's going on in your water. And this helps you figure out where you have sewage impacts, or at the very least, it helps you figure out where you have elevated nutrient levels, and you can start to figure out the timing of those levels and maybe get to something along the lines of a broad scale picture of what you need to do. But you can also have follow-up phase 2 targeted monitoring that helps you know where those sewage impacts are coming from. So that helps you figure out if you actually have sewage versus something else. And I'll talk about these towards the end of my talk. Nitrogen isotope techniques or other type of isotope techniques, pharmaceuticals and more advanced types of tracers, and then dye tracers. So first, those phase 1 tracers, that's what people usually are doing. That's what a lot of groups are doing. So nutrients there require something like an autoanalyzer or lab facilities. It usually runs for about $50 per sample, and you can get inorganic or total nutrients. Fecal indicator bacteria, when people do this, you can get a standalone system that can be operated by a citizen science group in that kind of order of magnitude. And once you buy some of your initial supplies, you're good for a little while. For physical parameters, you can use a sonde like a YSI or similar, and the range of those is all over the place depending on what you want out of it, if you want to do continuous monitoring or not, or which probes you want. But dissolved oxygen and pH would fall under that category. And then for that phytoplankton indicator, the chlorophyll A, you need filtration equipment and lab equipment. And if you're sending it off to a lab, it's generally around the range of around $20 per sample. And these numbers are all in US dollars. I just wanted to give you guys a range for like what those initial tests look like. And I'm assuming some of you or many of you have played around with one of these parameters and tried to figure out what that looks like. In practice, when you go out, it looks like this. On the left, you have our helpful person there holding a YSI, and on the right, you have bottle samples that are collected to try to do TSS or nutrient analysis. And then Hui Kawaiola here is the group on Maui, and they're working out of the back of their car with a turbidity meter there and other physical, another type of sonde, and they're showing you what it ends up looking like. So you can do this in the field. You can get the supplies to do it, and it can give you some baseline information. When you work up all the data, here's two different examples. A shout out to Surfrider here in Hawaii. This is their Maui Blue Water Task Force. They have their data online, and they measure for Enterococcus and can show where you have bacterial levels that exceed our Department of Health levels. On the right, you have kind of the results from that Hui Kawaiola group that are measuring for nutrients at all the different sites on the left there. And if you look, the highest one is Cove Park, which is next to a wastewater treatment plant and an outfall there. And so you have the data and it ends up working out like this, and this is what you can share with your communities. And I have the websites on there if you want to see most of that open source data. So the strengths then become, if you're measuring for nutrients with it and all these different phase 1 parameters, you can engage your community and become strong advocates, because the data can be interpreted and collected by the community itself. The data can motivate follow-up studies into source tracking and help identify big patterns. If you know where your low salinity areas are, you might be able to understand connectivity better. It creates a baseline over time. So if you do these for many years, you can see if there are changes due to new development or lack of agriculture, which is something we have here. Or you can start to correlate all of the data to coral health patterns and percent cover, so that you can see if the coral is responding to the different types of water quality that you have in wherever you are at your community level. And some of it's cost efficient, not always, but you can ask me about that in the end if you want to know what each of these things end up costing as a program. But there are limitations to every one of these methods. So for nutrients, they could be from either wastewater or agriculture, or it could be historical, it could be stormwater. So you can have high nutrients from a number of different sources. It's not going to necessarily tell you wastewater. Fecal indicator bacteria like Enterococcus are known for having issues in tropical places because it also lives in the soil. It's not an exact indicator that necessarily ties directly into wastewater. And there are lots of our colleagues who are working to improve this, but it's not good for just wastewater necessarily. For physical parameters, you can have low dissolved oxygen, but you might have other concerns that are not necessarily wastewater. And same for chlorophyll A. It's kind of a generalist. It's not necessarily going to say wastewater. So then how do you incorporate that previous data, which is easily taken by kind of the community science or an early set and move on towards phase 2? So I'll talk about our work in Pu'oko. That's the community in West Hawaii. They have 49 cesspools, 66 septic tanks. They have aerobic treatment units and 21 homes where we don't know what the OSDS is, all in that tiny little strip of land right next to the ocean there. Very, very close, right on the ocean. And it's a pretty popular tourist place with a healthy coral reef. But in recent years, our Department of Aquatic Resources has shown a decline in coral cover, an increase in macroalgae, and we've seen issues with coral disease. So this is an ongoing issue that's been studied for the last 15 years. And you can see that with the coral disease here, that the studies that have gone on have spanned that amount of time, kind of teeing it up and saying, hey, we have water quality issues, and starting to connect it to sewage, not knowing exactly where that sewage was. So in a timeline then, if this early research that is showing a decline and saying, hey, we think this disease and everything is an issue, from there you can do some of that phase 1 water quality work, and that was a consortium of Cornell, TNC, UH Hilo, and then follow on more advanced studies like nitrogen-15 isotope testing, microbial source tracking projects that become more advanced, and then a physical dye study, which I'll talk about in just a second. And all of that together, after all of those years, can be incorporated into a wastewater upgrade plan, which right now is being led by Coral. And then long-term monitoring can be continued in that phase 1 vein by Hawaii Viola to see if those upgrades are going to matter in the end. So this is sort of a timeline of what Pu'oko has looked like over the last 10 to 15 years. I want to just very briefly show you a sewage indicator score, which is something that the Nature Conservancy helped with to kind of lay out. If you have those phase 1 indicators, right, your nutrients, your bacteria, and then maybe something like isotopes, you have all those different layers, you can then start to isolate them and come up with a composite score that will tell you where exactly do I have the worst possible wastewater inputs. And so on the right there, you see that composite coloring with the hot areas being places where it's likely wastewater inputs that are causing elevated nutrients. And on the left, you can see some of the enterococcus levels as one of the data points that went into that method. But from there, if you looked at that timeline, you'll see that the UH-Hilo and some of the other partners then continued on to more advanced studies. Nitrogen isotopes is one of those phase 2 indicators that can help isolate whether it's a wastewater or agricultural problem. And nitrogen isotopes work by having any kind of nutrients that with nitrogen that go through a wastewater system might become denitrified, right? Their nitrogen is changing in form and gets this different isotopic composition. So you can get high nitrogen-15 isotopes when you have wastewater and low nitrogen-15 isotopes when you have fertilizer inputs, and therefore it allows you to distinguish places that might be affected by wastewater. The benefit to this is you can collect ulva tissue, so macroalgae tissue, and this is what Dan will be talking about after me, so I'll let him describe a little more. But that's one technique. Another technique that we have is to be able to use pharmaceuticals or other types of novel things that end up in our wastewater. You can use Tide, for instance, like there's lots and lots of things that anything that goes down your drain, if you want it, it will eventually go through your wastewater system. And there are usually techniques to be able to pick up on that. Pharmaceuticals is one of those. If you take an antibiotic or ibuprofen for pain or something like that, it's going to go through your system and not all of it will be absorbed and a lot of it will end up in the treatment system. And then you can measure it in the water. And we have techniques now to be able to measure it that are relatively cost efficient at $10 a sample if you're doing a certain number of samples. And the study I'll show that kind of goes through this, Mauna Bay, this is the study on Oahu. This site here had gone through similar process as Pu'oko, right? So we had early researchers say, okay, we have nutrient problems. We have the Department of Health that says there's elevated enterococcus. We have Surfrider involved on a regular basis. We have all of these different groups that are saying, hey, it's more and more and more complex to try to identify where the wastewater inputs were. And last summer, we led a project with HIMB, Hawaii Institute of Marine Biology, and Malama Mauna Lua, the community group that's working to improve the bay here, to kind of do a comprehensive study that added all the pieces together into a single snapshot. And we measured water quality at 178 locations. The little yellow dots there are all of the cesspools or on-site disposal systems. So you can see they're quite extensive throughout the entire watershed. And one of the techniques we used, in addition to measuring all of those phase 1 parameters, is going through and using a suite of antibiotics and glyphosate, which is an herbicide that would indicate for stormwater or urban stormwater runoff. And so these different types, the sulfamethazole, all these that are hard to say, ibuprofen, carbamazepine, are all different pharmaceutical drugs that people take that end up in the wastewater. And then you can use them specifically at different half-lives, meaning they stay in the water column for different amounts of time. So either 28 days, ibuprofen is the shortest, carbamazepine is a little bit longer, and the glyphosate can stay in the water for a really long time. And so it ends up looking like this. If you look on the bottom panel there, the warmer colors indicate places where you have ibuprofen hotspots. And those places are places where since it only exists for a couple hours to a couple days, those are places that very recently have connectivity to some sort of wastewater source. And then the figure above with the sulfamethazole shows a little bit longer term. Those samples can stay around for 30 days. And so this is what appears in the water column later. And then it can get even more. So now you can start to see, as the drugs degrade less fast in wastewater treatment systems and in the coastal zone, you can see the extent of where they end up being detected until you get to the fourth panel on the upper right, the glyphosate here. And that shows you kind of an extensive pattern of many, many samples. Upwards of 90% of the samples are showing that particular herbicide. The last type of technique for tracing that I'm going to talk about is dye tracing. And dye tracing is really, really visual. So you put a bright dye into your wastewater treatment system directly, either into your cesspool, into your septic tank, or into your wastewater treatment plant. And then you try to detect it downstream, either visually or using some sort of equipment. And this is a chemical tracer that you can use. And it's great in a place like Puako because it really showed that there was a time delay of hours or days between when you flush your toilet and when it gets into the ocean. And you can see in this picture that they were able to detect it pretty quickly off the coast. And it's pictures like this that can really motivate the community to respond when they realize how quickly and how connected the systems are. And so I'll leave you on that one to go and try to follow up with some of the resources there on how you do it. And I also want to highlight that within the last 24 hours, this was released. I haven't mentioned the USGS as a partner necessarily, but they do a lot of our groundwork here, and the most advanced methods come usually from them to be able to isolate problems. And they just put out this scientific report showing exactly what I'm talking about in their approach to detecting and identifying sources of wastewater in Hawaii. And so there's great methods in there for how to do site selection and all of the different compounds you can use for source detection at the end. And with that, I just want to thank the many, many people that have contributed to all of the research that I described, and present to you an email address for you to ask me further questions or in the chat. Thanks.
Thanks, Kim. That was great. We'll hear from Dr. Dan Amato next. We'll give him a moment to get his slides up. As a reminder, Dr. Amato is from the University of Hawai'i and will present about statewide efforts mapping impacts of sewage pollution, all to guide a cesspool removal process. Over to you, Dan.
All right. Thank you all for joining us today. I'm going to focus this talk on describing the statewide assessment of wastewater intrusion into the coastal regions of the Hawaiian Islands. Today, my talk is going to begin with discussing some of the wastewater sources and briefly touch on impacts to the reef. I will reintroduce Seaweed as a bioassay. Thanks, Kim, for briefly touching on that. And then I'll get into this wastewater study. We'll talk a little bit about our methods, talk about the preliminary results that we have as we are still kind of actively analyzing the data as it's coming in from the lab, and summarize what we know so far. So in Hawaii today, we have two main sources of sewage to the nearshore waters. One that we haven't talked yet about today is injection wells, and these are, pretty much deep holes in the ground where we inject like our gravity feed, treated sewage. It could be preliminary treated sewage, it can be, just primary processed or secondary treated sewage. And there are many large municipal injection wells in Hawaii, such as the one that I'm showing here in this image from Kahului. This is an injection well right here, one of many on site, and the wastewater goes into this well literally a stone's throw away from the ocean, right into that kind of sand about 100 to 300 feet down. And this facility is about 4.5 million gallons a day, and there's about 3 of these large facilities on Maui alone. Another type of injection well, which is more common in the Hawaiian Islands, are kind of private injection wells, and these are typically used in condominiums like the ones shown here in Ma'alaia on Maui. So each condo would have its own mini system where they kind of separate the solids and the liquids go down about 80 feet into the ground, again, right next to the ocean. So all these fluids are eventually making their ways onto the reefs. There's about over 100 of these in the state that we know about. And the second major source, which we've touched on briefly, are OSDS, or on-site sewage disposal systems. There's a couple different classes of these depending on the type of treatment. There's around about 110,000 statewide that we know about, and about more than 88,000 of those, about 80% of those are cesspools. And again, a cesspool is similar to an injection well in the sense it's basically a hole in the ground, but it's a very shallow hole. As Kim mentioned, they can be found quite close to the groundwater table and have easy access to that groundwater, and then flow along those flow paths towards the ocean. And so these OSDS, and particularly cesspools, discharge a lot of nutrients, a lot of nitrogen, a lot of phosphorus, and that's because there's really, like, you know, literally almost no treatment. It's raw sewage going straight from your toilet, going into a hole in your yard, typically. So this just has the full load of nutrients that's associated with that wastewater. And it's these nutrients that are really the issue for our reefs. It's been shown by, you know, studies around the world that nutrient loading or adding nutrients to reefs can be very damaging. Now, keep in mind that these kind of tropical systems, these tropical reefs, evolved in a very low nutrient situation. The water is so clear out here and in many tropical locations because there's generally no nutrients. There's not a lot of growth. There's not a lot of phytoplankton in that water, so the water gets really clear. Now, when you add even small amounts of nutrients, it can really change the reef dynamics. It can change the communities that live on those reefs, both attached to the surface and not attached. So this can damage corals. It can cause disease and lead to bleaching. It can change the microbial communities of these reefs, which has, you know, kind of cascading issues that we're still learning about. It can cause phytoplankton blooms, bring in opportunistic invasive species, and this can become an issue for even things like fish diversity. For instance, if you have a lot of these invasive non-native seaweeds that grow or algae that grow on the reefs, and you end up with a situation like you see in my image at the top from Kaneohe Bay a few years back. The reef is covered with these, you know, alien seaweeds that the fish don't like eating, and if there's no, you know, good food on the table, they're not going to stick around for dinner. So it can really change fish dynamics on the reefs. Again, it can cause these macroalgal blooms or these seaweed, you know, growth events where they can take over reefs, detach, wash up on the beaches. It's not a fun thing for folks that like to enjoy beaches. And I'm sure some of you have seen this happen in Hawaii and around the world. Another item is that these kind of massive growths of high nutrient water, algae growing in this water, storing that nitrogen, it has been shown to lead to turtle tumors, and that's something that we were experiencing, unfortunately, for quite a while in the Hawaiian Islands. I believe it's gotten a little bit better, but yeah, it's still another kind of nutrient loading issue here in the Hawaiian reefs. So we can use these algae and these seaweeds as a bioassay and to actually indicate the type of water that they are exposed to. So I feel that they are ideal bioindicators of water quality because these algae integrate these nitrogen sources, these nutrients, over time. They're typically attached to a rock or another surface, so that makes them easy to collect. They're easy to culture or grow in the lab, and they generally have low analytical costs. But more importantly, that the algal tissue, that seaweed tissue, reflects the nitrogen content of the water that they are found in. And so if you look at my graph here, as in the lab, as you add wastewater to seaweed in the lab, as you incrementally add the wastewater, that algae will suck up that nitrogen and store it. For these green, you know, seaweeds like Olva, that is stored as chlorophyll. So you'll notice my algal picture at the top of the scale here is very vibrant, huge, and dark green, and that's what you'd expect from a seaweed like this growing at, you know, in luxury levels of nutrients, versus one that is kind of in its ambient natural state on the reef. It'd be fairly translucent and have a kind of a smaller stature, lower growth rates, etc. And as Kim briefly mentioned, we can actually use the isotopic signature of these seaweed tissues to indicate the source of that nitrogen. And that works like this, we can see as you add wastewater to, again, in the lab, adding wastewater to these algae, they quickly take on the isotopic signature of the source. So in this case, this was wastewater from a facility in Maui, and so very quickly, with just around, between 10, 15% wastewater in that beaker, we find that this alga is reflecting that isotopic signature of the source, which was around between 25 and 30. And we can use this, for instance, in this way. Say we are collecting seaweed off the rocks on Maui, and we take the alga, we dry it, send it into the lab, and we get a value back of about, let's say, 3 here, as I'm showing with my arrow. The source is kind of ambiguous at this point. It could be fertilizer, it could be soil nitrogen. Looking at this graph, it could be a mix of wastewater. We're unsure at this level. However, if we have a higher value, let's say it comes back as 13 or so, we're fairly certain that the nitrogen in the seaweed tissue came from a wastewater source, a denitrified source, and that could be any OSDS or injection wells, etc. So we've used this around the state of Hawaii in the past, and so one kind of study I'll highlight here briefly is something that my research team published earlier this year, where we gathered 3 different species of seaweed around the island of Oahu. And so this figure here shows the island of Oahu with this color gradient, this heat map showing the density of OSDS. Again, think 80% of these are cesspools. So you can think of it as a cesspool density map if you wish. And what we found was that everywhere we collected algae in the vicinity of these high density areas of cesspools and injection wells, the delta 15N value of those seaweeds reflected those sources. So we had high values where we expected to find wastewater, essentially. And you can see this in the west side in Nanakuli and Waianae. You can see this in Waialua, where there's a municipal injection well and a lot of OSDS. On the North Shore, here we have kind of private injection wells and more cesspools. On the east side, Hauula and Ka'ava had very high values for delta 15N in all these seaweeds. In Waimanalo, and even here in Honolulu at Black Point, where there's quite a bit of cesspools in the Diamond Head area, and also in Ewa Beach, again, pretty high density of OSDS there. So we're using this as kind of a tool to detect wastewater in the ocean via these algal bioassays. And so, the state of Hawaii is not, this is a very known issue. And a few years back, the state legislature made some pretty progressive legislation where they stated that all, through Act 125 in 2017, that all cesspools need to be upgraded by 2050. And this is an excessively large task. If you remember the number of them, 88,000 plus that we know about, probably another 25% more of those we have not documented for one reason or another. And the legislature also asked DOH to evaluate residential cesspools, develop a report, work on possible funding options for upgrades, and the following year established a cesspool working group, which Michael Mezzacapo will touch on later today. And for more importantly to us, commissioned a statewide study to study sewage contamination in the nearshore environment. And so that's kind of what brought on this current study that I'm going to talk a little bit more about now. And so again, the goal of the study was to determine which coastal areas in Hawaii were most impacted by wastewater, and through the lens of trying to assist the state to prioritize areas for cesspool upgrades. And so we did this kind of following 2 different methods. We used a wastewater modeling where we were kind of tracking nitrogen using groundwater flow modeling, and also again coastal sampling of macroalgae and water around the state on 4 different islands. So for the wastewater modeling, we had two parameters of interest. There's wastewater nitrogen flux. So this was the kind of the amount of nitrogen that we expect to find coming out of the coastal environment into the ocean that's wastewater derived. And also coastal OSDS density, as we understand that high density of OSDS near the coastline will likely put a fair amount of wastewater to that coastline. And so for coastal sampling, we selected sites based on our preliminary nitrogen flux models. We chose 2 kilometer kind of swaths of coastline to do a high resolution sampling for algae and water. We collected between 5 and 6 algal species shown at the bottom of the slide on 4 main Hawaiian islands. We submitted the tissues to the UHM Biogeochemical Stable Isotope Facility. And from that facility, we got 2 major parameters of interest: the delta 15N value, which you've heard a fair amount about by now, and actually the percent nitrogen in those tissues. And so just to clarify, the delta 15N value will give you an idea of the source of that nitrogen, but not the amount. And so the percent nitrogen in the tissue actually gives you a better indication of the amount of nitrogen in the coastal environment. So those two they're linked, but I just want to kind of make sure you don't think that a higher delta 15N value, it does not mean that that you have more wastewater. We get that information from percent N. And we also collected coastal water samples, and I won't spend a lot of time on this today. Those results are still trickling in from the lab, but we analyzed the water for nutrients, nitrogen, phosphorus, silicate, and the delta 15N of the nitrate in that water. So I can share some preliminary results. Again, they're still in the very kind of exploratory phase of the results. We're beginning to write them up, but I can share this with you. So this is the results of our modeling effort for the 4 coastlines. This is the wastewater nitrogen flux modeling shown with the colored bands and dots around the perimeter of the islands. Also shown in blue stars is the coastal sample sites for algae and water. We collected over 350 algal samples, and roughly about a third of them show signs of wastewater derived nitrogen in those tissues from their delta 15N values. Another kind of preliminary results here. This was something that was kind of published recently by Michael Mezzacapo in a white paper. And we can see, if we look at the left image here, this is an experimental site on Maui and Wailea. We have an average delta 15N of around 6.9, which isn't a very high value, but compared to the South Maui control, where it's 2.6, that's significantly higher, and we would say that's definitely picking up on a wastewater signal. And if you look at the percent N, the percent N is also much higher in the experimental site versus our control. So this is kind of how we look at our data on a site-by-site basis. So in summary, so far we found that sewage is definitely a major source of nitrogen to the Hawaiian shorelines, and that the groundwater modeling is quite useful as a kind of a large-scale tool. But the seaweed bioassays are actually better able to detect these kind of fine-scale differences, especially when you can sample at a very fine scale. The algal data generally agrees with the model results, where we expect to find large amounts of wastewater. We are finding those signals in the algal tissues. But basically, data from both of these approaches, the bioassays and the models, should be used together to assist with the prioritization of areas for cesspool upgrades. So with that, thank you for listening. I want to thank the Nature Conservancy, the collaborators on this project, Celia Smith, Robert Whittier, Chris Schuler, Megan Daler, and Stephen Colbert, as well as our field and lab assistants. And I'd like to also thank the state legislature and you, Hawaii taxpayers, if you're out there listening, for helping to fund this study. So thank you all. And yeah, I would love to take your questions at an appropriate time. And yeah, I'll hand it back over to you, Kristen. Thank you.
Thanks, Dr. Amato. Appreciate that. We'll go ahead and hold questions for just a minute more. We'll hear from Michael Mezzacapo from Hawaii's Cesspool Conversion Working Group. And yeah, take it away, Michael. I will just say there's been a couple questions about sharing slides, and just a quick reminder that we will be sending out an email later today or tonight, depending upon your time zone, with the webinar recording. We're happy to share PDFs of the slides and the presenter's contact information as well. So yep, those will be coming to you. Thanks, Michael.
Thank you, and thank you to Dr. Falinski and Dr. Amato for those great presentations. So I'm going to kind of tie it all up. How do we use this fantastic science and community sampling efforts to help us make great policy, right? So on the left is a picture of the infamous cesspool that we've been talking about. So in 2017, not only did we mandate the conversion of cesspools, but we also banned new construction. So we wanted to make sure that because up to that point, we were still putting these in the ground, even though we knew that they were problematic. So then in 2018, the state legislature, they passed a bill to create a cesspool conversion working group. Okay, and it's a group made up of a diverse group of stakeholders, many who aren't scientists, but they were kind of tasked with addressing the problem from a systems perspective and come up with a comprehensive plan to convert cesspools statewide. But that's a challenge in a unique tropical environment that's also separated by the, we have many different islands, and they're all diverse culturally, climatically, geographically, and economically. So it's a monumental task. And so since the inception of the Cesspool Conversion Working Group, there's been a number of technical reports that we commissioned, and they were looking at what's the best available science. And so they analyzed things like pollution monitoring techniques and indicators, the technologies, so the actual like on-site systems, financing, and different risk analyses and methods to track those. And we also looked at what other states have done to combat this. So we didn't want to reinvent the wheel here. We knew that there were some other states struggling with cesspool issues, and so we wanted to kind of see what worked for them, what didn't. And we heard about some of those from previous webinars at Long Island. So I really think this quote speaks volumes. So, and most people I think would agree that you can't build good policy without good information. So in science is the vehicle that really helps us reveal the problem. Science makes the environment speak. So environmental policy needs that comprehensive scientific research in order to be successful. So the presentations we just previously witnessed, those will help us build appropriate environmental policy. But also we need to know that science is not really geared towards policymaking, right? Policymakers have to make decisions, and those decisions often occur whether or not the science is complete, because we have to act, right? So where science is seeking to prove or disprove hypotheses, policymakers are typically reviewing evidence to assess what's known about a specific situation at any given time, and then make that. So, but the Cesspool Conversion Working Group really is implored to incorporate an adaptive policy platform. So in the comprehensive plan, we need to allow for new science or updated science to be incorporated into new policies for future decision making. And one of those is strategic adaptive management as a vehicle to do that. And so there are several reasons why adaptive policy mechanisms might be useful. The first is there's uncertainty in future conditions, right? We don't know what's going to happen with sea level rise or climate change. So additional policy instruments, also maybe non-technical or social science alternatives, could supplement or substitute say, technological advances, and those can change over time, right? So we need to adapt to those. The third one is improving information equity between the regulators and the stakeholders. And so that means we can lead to alternative solutions and more transparency in the policy-making process, right? We get to see how the sausage is made. And finally, there is missing capacity for research or implementation at the beginning of the time of policy creation, right? So maybe we don't have the monies to do the research, but if we incorporate an adaptive method, adaptive management, then we can possibly make up for that in the beginning. So adaptive management is an approach where policies are treated as hypotheses, right? And management actions are treated as experiments. And essentially, it's a structured process of learning by doing. And the best way I can visually explain that is by this adaptive management cycle, right? So we plan, we do, we monitor, we learn, and then we repeat the cycle. And within those there's some small inner learning loops, and where you can make tweaks to those. So in inner loop learning one, we can make small changes to the strategies and plans, and they can be made in response to, say, learnings between the major planning reviews, right? And so this is all meant to be repeated once we get more information. And so large, complex problems, they often contain multiple mini loops, and they operate at different scales as well. So we need to understand this in action, right? This plan, do, monitor, and learn. And we've seen some of those monitoring efforts that Dr. Falinski highlighted, right? So if we have statewide citizen science monitoring in place, we can hopefully incorporate that into the policy making process for this adaptive management framework, right? And we also need to learn and do. So if we're doing the samples like Dr. Amato was talking about, doing the delta N15 and percent N for the algal tissues, right? We can figure out, oh, we need to make a tweak here and we should upgrade these systems faster, maybe because we're noticing more wastewater pollution. So it also comes in terms of like adapting to sea level rise. So maybe we can also talk about how we're permitting systems before we're even approving an on-site system in an area. So within our counties, the county levels are responsible for the planning, right? But the statewide efforts are the ones who control the on-site wastewater permitting process. So they need to talk to each other. And so that could be done in the planning phase, right? Or that could be something that we learn along the way and implement then in new policy. So that could mean that we adjust the statutes so that we can protect homeowners, coastal areas. We can then, like I said, monitor those pollution levels or number of homes at risk, and then make adjustments to those policies. And with that, I'll wrap up my presentation and join the group for questions. Thank you.
Thanks, Michael. Yeah, and thanks to all three of our speakers. That was really interesting and informative. And now like to open up the webinar for discussions and questions with all of you. TNC Senior Scientist Dr. Stephanie Weir will also be joining our discussion. Hi, Steph. Remember, you can either send us your question, comment, or example via the question box, or raise your hand and we'll go ahead and call on you and unmute you. And yeah, please just please share examples of your water quality related work too. We're here to learn from each other. Let's see, a number of questions have come in during the presentations. I'm gonna go back to the beginning and ask a question. I think this is directed at you, Kim, but how long does rhodamine dye stay in the water?
Ooh, that's a good question. I think I might feel that out to my Hilo partners. I know that we said they were on the call, but if you don't mind emailing me, I'm pretty sure that is a day to hours long dye system, but I would like to be able to second guess that since that wasn't my study personally.
Okay, so we'll keep that answer for now and see if any other answers come in via the chat. We had a question, so I'm going to jump around, but we had a question about your the adaptive management cycle, Michael. And the question is, what is the general time frame for delivering scientific information to the policymaker, seeing a policy pass, and then be applied to a community? I think that's a bit of a loaded question, but the general time frame of the cycle that you shared.
Sure, I think it depends on the scale of the problem. And since we have just sort of begun this effort and are still building this comprehensive plan, it's difficult for me to say in relation to our issue here in Hawaii. But I can say that, you know, adaptive management is being used in places like around the Great Lakes. So the International Joint Commission, for what they also they have a water quality agreement among all the states in Canada, and they use adaptive management policies. And so I think, you know, it probably takes anywhere from, you know, a few years to maybe a decade, depending on what's going on. It just depends on the problem and the scale of the problem. So that's a bit difficult. We're hoping to move a bit faster so that, you know, we have some of this new emerging science. Is there a way to input that a lot quicker? But we also want to make sure that it's a valid process and that it's delivering appropriate results, right? We don't want to jump on something new and experimental, make a policy, and then have that fail. So these issues take time. It's been seen, you know, in Long Island, it's taken decades to help them convert some of their cesspools and get going in the process. And this is a big ship to steer.
Great, thank you. I do see a hand up. I'm going to go ahead. Jason, if you'd like to ask your question or share your comment out loud, please do. You should be unmuted. Hi, Jason. I think we can hear you now. Oh. We'll give him a moment. We'll move on to the next question, see if he pops in. Is the cost for converting cesspools the biggest challenge of doing so? There's some thinking.
Yeah.
It's one of the biggest challenges, right? So in Hawaii, we're very isolated. And so it's also a challenge to get materials, to have materials manufactured here. So it's, you know, there's numerous challenges, and cost is only one of them. It's a big one, don't get me wrong. But if we had a source of money, it would certainly be less, right? But it's yeah, there are multiple challenges, and I think they all combine to make it a very large and complex problem.
Yeah, I'd like to add to that. It also comes down to location and you know, the geology of the area the cesspool's in, the depth to groundwater, the size of the lot can play a large role. I mean, you know, it's not as easy as let's just upgrade everybody to septic leach field, which isn't much better. But for a septic leach field, you need a fair amount of land that's open that you can dig into easily, that has, you know, depth to groundwater. So there's a lot of considerations with location and the geology of the location that need to be considered, you know, when deciding what is the best way to go for upgrades.
Can I ask you to just elaborate on that a little bit more on the general answer, and maybe switch gears and forget about cost and say, what are some maybe social challenges or political challenges that might be involved, if there are any, and if those are significant, or if there really is more of a logistical and cost challenge?
Well, in terms of creating an outreach plan so making sure that people understand why cesspools are a problem is a big challenge, so that's a social issue. Also connecting the levels of governance so that we're working in concert to address the issue. You know, one organization may be in charge, like Department of Health is in charge of the permitting process for on-site systems. But like I said, the counties are responsible for individual planning efforts, right? Creating subdivisions. So making sure that those efforts are in concert. So we're like Dr. Amato was just saying, let's say we know an area, you know, Department of Health is going to do a permitting review process, but let's say we know that you want to have a high density amount of septic systems in an area that's adjacent to the coast. Is that truly the best thing to do? And that takes planning and cooperation between those entities, and also homeowner outreach, so people understand, oh, you know, even if I have a septic and I live right on the coast, I may be contributing to ocean pollution and damaging the reefs. And so kind of helping people understand those links are very important and very challenging.
Let's see, and a ton of questions have come in. I'll do my best to get through some of these. But one question is just about how can nonprofit groups who want to do citizen science get involved?
One place you might be able to start is the National Water Quality Association, which does I think it's National Citizen Science Water Quality Association, which can help to provide all the early methods that you need. It helps to have a partner who has done this before to kind of help with site selection and all of kind of those things. And then there's a fundraising aspect and stakeholder building aspect to this too. So if you're a nonprofit that's just starting out, trying to identify the people in your community that might be kind of the spokespeople for this in the beginning, and then partnering up with one of the water quality monitoring organizations. It doesn't have to be in your home country or your home place, but there are lots and lots of people who have done this before. So reaching out and trying to figure out, how can I get those methods, right? Don't start from scratch, so that'll be a big step in getting it to the next step, which is kind of practicing and working, getting community members trained up, however that might be. But that would be some of the steps I would say to go about it. But there's a lot of support out there, and I'm happy to connect people if they ask.
Kim, I'm thinking too, we should share the link to the Huio Kawaiola, the Citizen Science Water Quality webinar that you helped us do last year or 2 years ago. It's a great overview of that program on Maui that Kim referred to earlier. It could be great. So we'll send that out with the email as well.
Thanks.
Okay, this question, there are a number of questions related to technologies and different solutions. Here's one of them. So there are a lot of new technologies that will enable areas that cannot be economically served by centralized sewage systems to move away from cesspools. Are the Hawaii planners looking at these technologies, especially small-scale filtration?
I can sort of answer that. So I'm not a wastewater engineer, and technology is not in my purview. But I can tell you that yes, the state has commissioned a technology report that went over many different emerging as well as proven technologies. So when I say that, I mean looking at things like aerobic treatment units, smaller scales, wastewater treatment systems, as well as some waterless options and composting, basically all the available technologies that could possibly be put in. They're studying those, they're evaluating them, looking at costs, manufacturing process, all that. But I can't specifically answer that. I don't know if, Dan, if you want to touch on anything more or.
Yeah, I'll jump in here. I'm not that familiar with, filtration microfiltration in general, but I would like to add that there has been a lot of energy and a lot of money and research dumped in specifically by the Gates Foundation to reinvent the residential toilet. And so I feel like that could really be that type of technology could really be a powerful thing for, you know, a lot of the types of residential units we have here in the state where it's a 3-bedroom, 1 bath. And so, you know, if you were able to purchase one of these units, some of them have literally no waste. They make, you know, they make clean water. They don't put wastewater into the ground. The solids that are generated can be used as fertilizer. So there's a lot of kind of byproducts that are useful, and in addition to the not contributing to groundwater pollution. So I think that we should kind of keep an eye on these technologies as they become more and more available to residents and consumers.
I'll add one more thing. There are nonprofits in the space, do local ones. There is VI, which is Wastewater Alternatives and Innovations, and they're looking at technologies that could benefit Hawaii and are good for Hawaii specifically. So that's just another partner that is involved in that.
Just want to add one last thing, that these technologies are being tested in Hawaii too. There's one of these, you know, new toilets at the HIMB as a pilot study.
Thank you. And we'll take one last question here. Dr. Amato, I'm assuming this is directed at you. Can you speak to the fact nitrogen fertilizer and sewage sources potentially cancel each other out in an N15 tissue value? Are the nitrogen values in tissues a potential way you are summarizing or differentiating overall nitrogen?
All right, let me just unpack that slightly. So I'm trying to understand the question. I believe you're asking, if you have multiple sources in one location, will they cancel each other out? And so I suppose that could happen if all of those sources had the same amount of nitrogen coming from them. And that could definitely lead to kind of the mixed signal that I was talking about in the lower value, something between, you know, -2 and 3 or something like that for the delta 15N. But this is where the percent nitrogen really becomes a useful tool used in conjunction with the delta 15N value. For instance, if you have quite a low value of delta 15N, like 0 or 1, but you have a very high level of percent N in that tissue, there's a graph that Kim actually showed earlier in her presentation that would help assist this. But that's a fertilizer signal. You have a fertilizer signal of delta 15N and a lot of nitrogen, which would indicate that you have a fertilizer source. Because you wouldn't have that if it was a natural source. You wouldn't have that much nitrogen in the tissue. So you have a high level of nitrogen, you have a fertilizer source. And so again, that signal, whatever the alga has in it, is this kind of mix of whatever it sees over the time it's been in the water. So if it mostly sees sewage-related wastewater, you're going to have a value that reflects that sewage. If it's a mix, you'll have a mix. It'll be more ambiguous. So it's not a one-stop shop for determining source. You have to kind of take the clues into account. You have to look uphill and see, do you have a wastewater plant? Do you have a large amount of cesspools? Do you have a pineapple field? Is there legacy sugarcane ag that was working there? So you have to use environment as a clue as well, and look uphill. Hope that answers your question.
I'll pass that message along. I hope that answers your question. We are going to continue, since we are at the top of the hour. We have a number of questions and comments that have come in, so we'd really encourage you to continue this conversation on the Reef Resilience Network Forum. So that's our online discussion platform for reef managers, practitioners, and experts. So to continue the conversation, and if we didn't get a chance to address your question, or you want to share an example or a comment from today's webinar, please go to forum.reefresilience.org/. We can put that in the chat box for you. And we will be, actually before I wrap, I just want to see, do any of the speakers want to share any final thoughts or anything you wanted to say that we didn't get to cover in the discussion? You're good? Okay. You'll see a quick survey at the end of the webinar. Please share your feedback on this webinar and on your interest in the topic of sewage. We're using your feedback to continue to build out this ocean sewage series. Our next activity in the series will be a virtual tour of the new sewage pollution web resources that I mentioned previously. These web pages are going to summarize existing sewage treatment systems and water quality monitoring methods, impacts on marine life, impacts on human health, emerging management solutions. So that'll really be a lot of information there that'll address some of the technology questions and alternative to cesspool questions that were coming in. And then some final pages sharing ideas for communication and collaboration to address ocean sewage pollution. So these new resources, along with a recorded tutorial that we'll be making, giving you an overview of the resources, will be released in January. So stay tuned, we'll be sharing information about that. And just want to say a big thanks to all of our presenters, and thanks to everyone for joining us today. Happy holidays, and we look forward to connecting in the new year. Thank you.
Thank you.